Hello students, today let us learn about the ultrastructure of chloroplast. Let us start with the introduction. Photosynthesis is the process which supplies the energy and reduced carbon required for the survival of all life on Earth, in addition to providing molecular oxygen necessary for the survival of aerobic organisms. Chloroplast is one of the most important organelles as it is the site of photosynthesis and is found in plant and algal cells. It is the most widespread forms of plastid which can carry on photosynthesis due to presence of green pigment known as chlorophyll. In the cells of algae and green plants, the chloroplast is largest organelle after vacuole and form the most vital organelle. In algae, they show great variation in size, shape and number. Their morphology in algae is so species specific that it is used as a criterion for taxonomic classification. They may be like a convex lens shape but may also be a spiral as in Spirogyra, star shape as in Zygnema or irregular shaped in Oedogonium. The algal cells may have only one chloroplast as in Chlamydomonas measuring over 100 micrometer in diameter, or there may be 100 smaller ones. The cells located in leaves of higher plants can have 20 to 50 chloroplasts. So, in higher plants, about 500,000 of chloroplasts per square millimeter of leaf surface area is reported. The shape of the chloroplast found in leaves is generally lens or oval shape with one convex face with the other face plane, convex or concave, and can be as long as 5 micrometer and typically 2 to 3 micrometer in diameter. The chloroplast does not occupy a specific position in a cell as they are in a constant motion because of cytoplasmic streaming. Let us now see the ultrastructure of chloroplast. Chloroplast was first seen under microscope by the plant anatomist and grew in the 17th century. However, the discovery of this important organelle is credited to Julius von Sachs, who is also known as father of plant physiology. The first electron micrographs of chloroplast were published by S. Grenick and K. Porter in 1947, showing their detailed ultrastructure. The ultrastructure of chloroplast shows that it has 3 compartments with an outer envelope, an internal lamellar membrane system and stroma. The outer envelope is double membrane system with each unit membrane about 8 to 10 nanometer thick. The outer chloroplast membrane is highly permeable to small molecules and ions, but not to large proteins. The inner chloroplast membrane borders stroma and is less permeable with many transmembrane proteins embedded on it. Due to its less permeability, it regulates the movement of molecules inside the chloroplast in addition to providing site of synthesis of carotenoids, fatty acids and lipids. A narrow intermembrane space of about 10 to 20 nanometer occurs between outer and inner chloroplast membrane. In addition to the above 2 membranes of outer envelope, there is an internal lamellar membrane system which forms a network of parallel flattened membrane bound system and forms a disc-like membrane sacs called as thylakoids. The term thylakoid, which means sac-like, was coined by Menke in 1962. The extensive folding of the lamellar membrane stack form the grana and it leads to increase in grade surface area. Because of these infoldings, a typical leaf has 600 times its surface area of photosynthetic membranes for its cell layer containing chloroplast. Each granum may consist of 2 to 100 thylakoids. The grana present inside the chloroplast are interconnected by paired membranes known as stroma lamella. The lumen of each thylakoid is connected with the lumen of other thylakoids, which defines another internal compartment called thylakoid space. The thylakoids contain the photosynthetic pigments, which include the chlorophylls and others like the accessory pigments like carotenoids. Chlorophyll and proteins bound to the thylakoids use light energy to make simple sugars from carbon dioxide and water. The substructure of the internal membrane system of chloroplast varies among different plants. Stroma or matrix forms the third component of chloroplast and is the large space surrounded by the inner chloroplast membrane. In the stroma, several kinds of discrete bodies are present, which includes DNA, RNA, oil droplets, ribosomes, proteins and other materials such as starch grains. Starch grains up to 2 micrometers in length lie freely in chloroplasts of higher plants or are associated with internal membranes. The starch grain is found in chloroplasts of green plant tissues that have been actively photosynthesizing. The main protein component found in stroma is ribulose bisphosphate carboxylase or rubisco. The ribosomes found in the stroma are smaller than for those found in cytoplasm and is of 70S type. Presence of plastoglobuli is a common feature of chloroplast. It is the reserved lipid molecules with a size of about 0.1 micrometer in diameter, which starts depositing when leaves change their color. They may be found in close association with granule membranes or they may be found freely in the cytoplasm. The DNA found in chloroplast is a circular molecule which is not associated with histone proteins. They occur in localized stromal regions called nucleoids, with the number of nucleoids differing in different stages in development of chloroplast. The chloroplast DNA is not autonomous, as many enzymes in the way of chlorophyll synthesis are located in nucleus. The gene for synthesizing large subunit of the important enzymes like rubisco is located in chloroplast DNA, although the gene for small subunit is located in the nuclear DNA, showing its semi-autonomous nature. Let us now learn about the photosynthetic pigments. During photosynthesis, light coming from the sun will enter photosynthesis through absorption by the pigment molecules present inside chloroplast. Pigments are the molecules with their own characteristic absorption spectra in response to light. These pigment molecules are necessary for photosynthesis and are known as photosynthetic pigments. The photosynthetic process depends on a set of complex protein molecules and pigment molecules that forms the two photosystems, that is, photosystem 1 and photosystem 2, that are located in the thylakoid membranes. Each of the photosystems has two components formed by antennae complex and reaction center. The antennae complex is a light harvesting complex formed by chlorophyll and carotenoids with proteins. They capture the solar energy and transfer it to reaction center formed by chlorophyll molecules. The reaction center of photosystem 1 has absorption maximum at 700 nanometer and that of photosystem 2 at 680 nanometer. The essential feature of photosynthesis is absorption of visible light coming from sun by the pigment molecules. Visible light is a combination of many different wavelengths of light which appear as white light and seen as different colors when it is separated. The color of the pigment comes from the wavelength of light reflected depending on the type of pigments. So, the colors of the photosynthetic pigments that we perceive also depend on the type of pigment and they absorb at different wavelengths of absorption spectra. There are three primary photosynthetic pigments found in plants and algae. These are chlorophylls, accessory pigments, and phycobilins. These pigments have their own characteristic absorption spectra which give them different color. The energy of sunlight is first absorbed by the pigments of the plant. All pigments active in photosynthesis are found in the chloroplast in plants and algae. First, let us learn about the chlorophylls. The most common type of photosynthetic pigments are the chlorophylls. Among the different types of chlorophylls, chlorophyll A and chlorophyll B are the more common and abundant ones, which absorb strongly in the red and blue portions of absorption spectrum and reflects green light. Because of this, chlorophyll containing plants are green in color. Chlorophyll A is the main photosynthetic pigment that participates directly in the light-dependent reaction of photosynthesis and is found in almost all the photosynthetic organisms. Chlorophylls are the key pigment molecules driving photosynthesis and is highly sensitive to light, but it may be destroyed by too much light. Chlorophylls have a complex ring structure that is chemically related to the porphyrin-like groups found in hemoglobin and cytochromes. This porphyrin ring is a stable molecule where electrons are loosely bound. As the electrons move freely around the ring, it has the potential to gain or lose electrons easily and thus the potential to provide energized electrons to other molecules. This is the fundamental process by which chlorophyll captures the energy of sunlight. Among the several kinds of chlorophyll, the most important and most abundant one is chlorophyll A. Thus, chlorophyll A makes photosynthesis possible by passing its energized electrons on to different molecules. Chlorophyll A absorbs light with wavelengths of 430 nanometer in the blue region and 662 nanometer in the red region. The hydrophobic or fat-soluble phytol chain is found to be embedded in a lipid membrane and the tetrapyrrolic ring is placed outside of the membrane. This tetrapyrrolic ring of the chlorophyll absorbs the energy from light. The metal magnesium, which is present at the center of the tetrapyrrolic ring, can have variable oxidation states. So, magnesium can accept and donate electrons readily depending on the situation. All green plants, algae and cyanobacteria which photosynthesize contain chlorophyll A. A second kind of chlorophyll is chlorophyll B, which occurs only in green algae and in the higher plants. This molecule has a structure similar to that of chlorophyll A. It absorbs light of wavelength 453 nanometer and 642 nanometer maximally. It is not as abundant as chlorophyll A and probably it might have evolved later. Other chlorophyll types are C, D and E, which are found in some protists and cyanobacteria. The photosynthetic bacteria use the bacteriochlorophyll pigment similar to the chlorophylls for photosynthesis. The chlorophylls and bacteriochlorophylls or pigments found in certain bacteria are the typical pigments of photosynthetic organisms, but all organisms contain a mixture of more than one kind of pigment, each serving a specific function. Next, let us learn about the carotenoids pigments. Carotenoids are found in all photosynthetic organisms which are integral constituents of the thylakoid membrane and they are also known as accessory pigments. Carotenoids are usually red, orange or yellow pigments and include carotene and xanthophyll. These compounds are composed of two small 6-carbon rings connected by a chain of carbon atoms. As a result, they do not dissolve in water and must be attached to membranes within the cell. Carotenoids can absorb photons with wide range of energy, although absorb light between 460 and 550 nanometer maximally and appear yellow, red or orange. Carotenoids pass their absorbed energy to chlorophyll as it cannot transfer sunlight energy directly to the photosynthetic pathway. In mature leaves, the presence of accessory pigments is greatly marked by the presence of chlorophyll, but during the leaf fall, when chlorophyll disintegrates, the reds, orange and yellows show through accessory pigments. Accessory pigments are also responsible for the brilliant colors of vegetables such as carrots, tomatoes, eggplant and peppers. Next, let us learn about phycobilins. Various species of algae and photosynthetic bacteria have other kinds of photosynthetic pigments not found in plants, which are known as phycobilins. They are water-soluble pigments and are found in the stroma of the chloroplast or in the cytoplasm. They occur only in cyanobacteria and rhodophyta. The two classes of phycobilins are the bluish pigment phycocyanin, which gives the cyanobacteria their name, and the reddish pigment phycoerythrin, which gives the red algae their common name. When phycocyanin and phycoerythrin are exposed to strong light, they absorb the light energy and release it by emitting light of a very narrow range of wavelengths. Phycobilisomes are supramolecular complexes primarily composed of phycobiliproteins, which are brilliantly colored family of water-soluble proteins that covalently attach to the phycobilins. Thus, phycobilisomes serve as the primary light-harvesting antennae for photosystem II in cyanobacteria and red algae. Coming to the conclusion. Thus, we have seen that chloroplast present in algae and plants is the site of photosynthesis which harnesses solar energy. There is a need to know the detailed structure of chloroplast as it is the only organelle which can perform photosynthesis. Among the three envelopes of chloroplast, thylakoid membrane system is very important. The photosynthetic pigment molecules which are located in thylakoid membranes will absorb light and convert solar energy into chemical energy. Among the mixture of photosynthetic pigments, chlorophyll is the most important, with chlorophyll A present in all photosynthetic algae and plants. However, due to accessory pigments present in addition to chlorophyll A, the photosynthetic organisms can absorb visible light in wider range, which will lead to improvement in their efficiency. Thank you.